Do you know that we consume food every day, trusting that it is safe, nutritional, or of good quality? But what if this food contains invisible toxins, harmful microorganisms, or even misleading nutritional content?
Wow.
Yeah, of course. And substances like the aflatoxins, microbial contaminants, and inadequate nutrient levels pose serious risks to human health.
Wow.
Of course.
As part of our research, we visited the food science lab to observe how these techniques are used in quality control.
So it's food microbiology lab again. We conduct microbiology tests on food samples. Okay, so commercial samples and then students' project results. Yeah. So I'll take you through some of the equipment we have at the lab. Okay. So this here is an incubator. It's kindly set to 25 degrees Celsius for microbes like yeast and molds. And this is an incubator shaker, mostly used for liquid cultures. And then this is also an incubator, and it's currently set to 37 degrees Celsius for aerobic phase counts. So this is a water bath.
And then at this lab, what we usually use it for is when we are done preparing the media, also cleaning, and then we have to do something else. And then the media has a temperature. When it gets solidified. So we keep them in most of the time to make sure that it's within the temperature that will still be liquid for you to pour when the time is due for you to use it.
By bringing it to boil, you boil using the mention that.
So my question is, how does analytical biochemistry help you? So how does food analysis help in quality control?
All right. So when it comes to quality control, we are looking at number one, the quality, and then the safety of the food that we consume all the time. So with regards to, especially when it comes to the food analytical side, mostly we are concerned with the shelf life, how a food is able to stay on the shelf for long without spoiling. And and so we determine the moisture content of such food, because this helps us to know the microbial activities that might, these microbial activities, they tend to lead to food spoilage when we realize that the moisture content is high and all that. So with this, we're able to know that this food is tend to spoil early or not. And when it comes to the fermentation or the microbial side, the number of counts we are able to estimate on food products will tell us that this food is safe for us to eat or unwholesome for consumption. Yeah.
Alright, so basically when it comes to quality checks, depending on the type of food substance or food sample that we're analyzing, it will tell us what kind of parameters we should look at. So for example, if we are looking at a food sample that was prepared through mechanical grinding and all that, for our health safety, we have to look out for some traces of heavy metal that might be in the food. Maybe the machine that we used wasn't good. So we have to do heavy metal analysis where we look out for two basic elements, that is the lead metal and then the arsenic. That is that, by the way. For most dried food sample, basically we are expected that there should be a new moisture percentage. So in order for us to see that this is a good product, we have to check moisture analysis moisture content on it. And then it is once we know that the moisture content is low, then it may mean that the dry product is good, especially dried powdered like the John Brown and the cereal mix and stuff. And then also we check for something we call acid insoluble ash. Now in every food product, there is a component of that food product that is ash content. So it is estimated that this ash content should be able to dissolve in acid. So that once taken, in our normal, we have acid in our stomach, and some of these things should be broken up or be dissolved completely. And so once you check that and it's very low, then it means that it is okay for people to consume. But when we check and it's very high, then it means that probably the food product isn't really a food product. It's made of more like sand or something like that. So that one we look out for that. But aside that, the main thing that we look out for are microbial parameters, where we check various microbes that might be in the food. So especially we look out for aerobic plate counts, enterobacterial size, staphylococcus, yeast and molds, salmonella, and a whole lot, bacillus, clostridium. These microbes, depending on the kind of food. For example, for every meat product, it is sure likely that we have to check for salmonella, because salmonella tends to play around meat products, especially the frozen ones and all that. So these are some of the tests and many more that we look out for depending on the type of product that we're making.
Thank you very much.
That is the spectrophotometer, which is used to measure the absorbance of light by a sample. This helps in determining the concentration of substances in food, such as nutrients or contaminants, making it a key instrument in analytical biochemistry.
First of all, we have to digest the sample. And what we do is we use acid and a catalyst to do the digestion. After we feed our sample in test tubes, in digestion tubes, we will add sulfuric acid and then some catalyst, like let's say selenium powder. Then we put it in this heating block where we will leave it there for some time. And once digestion is complete, then we add we now distill. But first of all, we dilute the sample, then we distill with our Kedal setup here. So this is where we do the distillation, and once we are done with the distillation, we mount up the burette and then the retort stand heat titration to be able to determine the titer value, which will be used in calculating the protein contents. Thank you.
From detecting microbial contaminants, analyzing the food to verify its nutritional content, and identifying harmful toxins that could harm our body, analytical biochemistry plays a crucial role by providing essential techniques such as spectrometry, chromatography, electrophoresis, microbial and biochemical assays for effective food quality control. It not only safeguards what we eat, but also ensures that what we consume daily meets the highest standard of safety and nutrition.